Hi, my name is Kesarin Mehta, and today I'll be talking about my UROP project focused on using AlphaFold 3 to explore protein interactions necessary for plant immunity. Plants are constantly exposed to biotic stresses, such as bacterial and fungal infections. Global food security is significantly impacted by plant pathogens and their destruction of crop yields. Roughly 20 to 40% of crop yields are lost each year due to plant diseases costing an estimated $220 billion annually. As the effects of climate change continue to contribute to food insecurity and hunger, finding safe and effective ways to improve plant immunity will become increasingly important. Similar to humans, plants have a set of immune responses that allow them to defend against invading pathogens such as fungi and bacteria. In order to recognize pathogenic microbes and mitigate infections, plants often employ a process called pattern-triggered immunity, or PTI, where small proteins or pathogen-associated molecular patterns originating from the microbe are recognized and bind to the host receptor-like protein, often located in the apoplastic space. After binding between the RLP and PAMP occurs, a signaling cascade, facilitated by co-receptor proteins with kinase domains, leads to the activation of defense-related genes and immune responses. RLP53 is a receptor-like protein from the land plant Arabidopsis thaliana and has been implicated in conferring increased resistance to various species of fungi, bacteria, and oomycetes. However, the exact mechanism and ligands involved in RLP53-dependent immunity remain unknown. Traditional methods for studying protein-protein interactions such as X-ray crystallography and NMR can be time-consuming, expensive, and require large amounts of protein. To date, only 3 RLP co-crystal structures have been solved. To work around these limitations, I use the computational program AlphaFold 3, which uses deep learning algorithms to predict and model the 3D structures of proteins. By comparing information from protein structure and sequence databases, AlphaFold 3 integrates coevolutionary information from multiple sequence alignments and pair representations to generate predicted protein structures and interactions with varying levels of confidence. This confidence is represented as a per-residue model confidence (PLDDT) or interface predicted template modeling (IPTM) score ranging from 0 to 1. Higher IPTM scores indicate greater confidence in the predicted interaction in reference to similar structures and tend to be strong candidates for experimental validation. In order to avoid testing all the possible proteins in the proteomes of pathogens known to be involved in RLP53-dependent immune responses, I chose 2 separate approaches to narrow my initial pool of candidate ligands. The first approach involved taking the proteome of the fungal pathogen Galliminomyces sicariceum and running it through the secretion prediction software signal P6. Based on the signal peptides at the beginning of each sequence, G. sicariceum proteins were processed and organized into categories based on their predicted probability of secretion. These secreted proteins were then analyzed with the program effector P3 to predict their likelihood of being effectors and localizing to the apoplast, cytoplasm, or both during host infection. From an initial proteome of 6,757 proteins, this pipeline narrowed the final pool of candidates to 173 predicted secreted effector proteins. These tools were critical because they helped filter thousands of proteins down to a manageable number of likely candidates, saving time and resources. My second approach involved using NCBI delta blast P to search for homologs of the cognate ligand IF1 from the closest characterized homolog of RLP53, RLP32. NCBI delta blast P works by identifying conserved domains in the sequence of the query protein from the conserved domain database to generate a point-specific scoring matrix, which is then used to search for similar proteins in the protein database. Since RLP53 and RLP32 are related, I focused on proteins similar to IF1, assuming that Arabidopsis thaliana and its pathogens may have co-evolved. IF1 is an S1 beta-barrel domain containing protein involved in RNA binding and translation, so proteins originating from known pathogens with RNA binding and translational functions were also included in this search. To the left, an AlphaFold 3 PDB structure depicts the interaction between RLP32 in green and IF1 in orange. After testing a total of 226 proteins from both search methods against RLP53 and AlphaFold 3, I generated a distribution of the IPTM scores for each predicted complex as a histogram. The cutoff values for identifying likely interactions was calculated as the mean plus 2.5 times the standard deviation, resulting in a threshold of 0.57. Google DeepMind, the developers of AlphaFold 3, recommend IPTM scores between 0.6 and 0.8 as a benchmark for likely interactions. This range is highlighted in green on the graph with the cutoff value indicated by the vertical black line. Notably, RLP53 and its highest scoring predicted ligand achieved an IPTM score of 0.74, comparable to confidence levels observed in experimentally validated receptor-ligand pairs from other plant immune receptors from AlphaFold 3 predictions. 4 total proteins, 2 from each search method, had IPTM scores above the 0.57 value and were selected for further study. The exact identities of the predicted proteins are not disclosed, as this research is part of an ongoing project and we aim to prevent premature dissemination before experimental validation. These results suggest that AlphaFold 3 may be a useful tool for discovering new ligand-receptor interactions in the context of plant immunity, acting as a potential alternative for initial screening. Future work will include experimental validation of these potential ligands to confirm their role in RLP53-mediated immunity. If validated, these ligands could provide insights into improving disease resistance in crops, which is vital for food security in a changing climate. I would like to say thank you and acknowledge Kaylee Richard and other members of the Sarkar and Katagiri labs for supporting this research, as well as the Department of Biomedical Engineering and the Office of Undergraduate Research UROP program for funding this project. Thank you for listening.